Thanks, Aaron. Hang in there. We're almost getting to the end. We're located in Gaithersburg, Maryland, and the company is MaxCyte. And what we're going to do today is, let me get my slides up. Great. Is to give you a little background on the technology, talk about some of our applications, and I'm going to spend most of my time talking about our approach in the CAR T cell therapy space and what we believe to be a really interesting, disruptive way that we're moving that area of cell therapy into the clinic. So I'm going to talk about, get this moving, sorry. Okay. So we are the leaders in flow electroporation. We invented this technology and have developed it. And the technology is based on electroporation, but it's not like the electroporation that you're probably using in grad school. It's a large volume system. It's a sterile, closed system for clinical purposes. And what we're doing is taking large volumes of cells and inserting molecules into those cells very rapidly. So we can process 10 to the 10 cells in about 20 minutes. 95% plus efficiency and 95% plus cell viability, a completely sterile environment. And the way electroporation works is we're taking a cell, we're disrupting the membrane very, very gently with electrical pulses, and then the molecules that are driven into the cell, and the molecule that is specifically engineered for that change in the function of the cell. And we're able to put in things like messenger RNA, DNA, small molecules, large molecules, all the way up to an artificial chromosome. So we have a lot of experience. Our systems are CE marked, FDA cleared. They're fully scalable. We can run the process from a few thousand cells all the way through to 10 to 10 cells, which happens to be the volume of a unit of 100 ml blood volume. And also we can run 2 times 10 to the 11 cells, which is the output of a 200-liter bioreactor in about 30 minutes. So it's a very novel technology, very useful technology, fully scalable, as I mentioned, high performance, consistent from run to run, and we have been able to work with basically any cell and any molecule. The work that we do is primarily in hard to transfect human primary cells. That's where we focused all of our attention, and I'm going to talk more about that in a minute. But talk about the applications for the technology. It's really in 3 areas. The first is in drug discovery, and this is where we're using large volumes of cells, human-like cells, to create assays. We work, for instance, with Vertex to create an epithelial cell that lines the lungs of cystic fibrosis patients, and using those cells to create assays to find new small molecules to hopefully have an impact on those patients. We also work with product development companies, and what they're doing with our technology is to produce large volumes of transiently produced proteins for manufacturing and also for early stage clinical trials. We have 9 of the top 10 pharmaceutical companies who use the technology, the top 3 in Japan. I spend most of my time, the remaining time, talking about cellular therapy, really in 2 areas. One is in gene editing, and the second is immuno-oncology. You'll notice the companies that are on that list are the leaders in the field. Many of them are in the audience, and many of them are at this meeting today. We work with all these companies. They use our technology to load molecules into the cells to actually do the gene editing. So, for instance, we work with Sangamo, and they use our technology to put messenger RNA to encode a zinc finger nuclease to either disrupt the cell or to insert a gene into a cell. So we work with all these companies, and we have quite a bit of experience. We have over 30 programs currently with clinical developers, half of which are in the clinic, and the other half are in late-stage preclinical trials. We focus our attention on an area that I know many of you are very familiar with. It's chimeric antigen receptor-based therapies. I don't think it's possible to overstate how exciting this is to not only patients and clinicians, but also to our industry, and how it's really helped to advance the programs and many of us in bringing investors into the space. And of course, this works by loading a molecule, a chimeric antigen receptor, with a recognition site that recognizes the antigen of a tumor cell. And then once it recognizes that tumor cell, whatever's tethered to it upregulates the T cell and then kills that cell. The CAR-19 process, this is the process that's used with lentivectors. So lentivectors or other retroviruses are used to transfect that cell. It's a almost permanent transfection, if you will, and so those cells are going after any cell that expresses a CD19 antigen. The great news is it's been able to wipe out and show incredible efficacy for leukemia patients and lymphoma patients, but it also wipes out any tissue that expresses CD19, of which B cells are also CD19 expressors. And so every patient that's on this therapy has B-cell aplasia and has to be on a lifelong IVIG therapy. Also, 1/3 of these patients have cytokine release syndrome, so it necessitates that this therapy is done either in or close to an ICU, and these patients are closely monitored for the period of time that it takes to ensure that they're past that point. From a manufacturing perspective, this process takes anywhere from 5, 7 to 2 weeks. The actual process of taking the cells, selecting and expanding the cells, and providing them back to the patient. It's obviously a complex procedure, it's time consuming, it's costly, and has to be done in a special manufacturing facility. And there's certain requirements for large scale production of the CGMP lentivector as well, which some of the companies here today have been talking about their approach to large scale manufacturing. So there's some challenges with taking this further into the clinic, not only for CD19 based drugs, but also another area that's very important is for solid tumors. And as folks have worked to move this into treating solid tumors, there's been some rather serious adverse events, and there's quite a bit of work now being done with switches and other strategies to prevent this on-target off-tumor toxicity, where it would actually, the tumor cell would express a certain antigen, but normal tissue also expresses that antigen. So we began working with Dr. Carl June and Dario Campana 7 years ago, and they approached us because they were concerned about a couple things early in those days. They were concerned about pediatric leukemia patients. It was one thing to give a patient who was 60 or 70 who had leukemia this therapy and lifelong IVIG treatments, but it was quite another thing to be giving this to a 7-year-old or an 8-year-old. And so they were concerned about having those patients on lifelong IVIG therapy. So they asked us to work with them and look at replacing the lentivector with the messenger RNA. And the idea here was that lentivectors express the CAR-19 for upwards of a month or even longer in most cases. And mRNA expresses the cell, the CAR molecule in the cell for just a few days, 3 to 7 days. And they thought that we could make the product from messenger RNA produce enough material that we could actually dose patients 5, 6 times on a subsequent dosing schedule. So this is a cartoon that illustrates the top cartoon shows the current the CAR-19 manufacturing process, which is basically done by almost all these manufacturers. And that's where you do the leukapheresis, you isolate the peripheral blood monocyte, you separate out the T cells, you then add the lentivector, you expand the T cells, and the lentivector needs to have the cells in expansion mode for it to transduce. And then after that's done, it's washed and aliquoted and cryopreserved. What we did at Penn and what we did at National University of Singapore with Drs. June and Campana was only change one thing, and that was we wanted to use our technology, which is a large-scale transfection technology, after the T cells were expanded. So they selected the T cells, they expanded the T cells, then they used our technology to put RNA into the cells so that we would have a similar cell population. And now we're working with 10 of the 10 cells that we needed to do this work. So we were successful in doing that. There's been 20 patients that have been treated using messenger RNA. These are solid tumor patients. There's about 150 patients that have been treated with CD19. So this MaxCyte approach, we've been very quiet in our approach to this, but we've treated more patients than any other approach as it relates to CAR and solid tumors, again with messenger RNA. And we've gotten no indication of any adverse events from this, so it appears to be safe. We're a long way from proving efficacy, but we are seeing some important signs. And these are presentations that were done at ASCO and AACR over the last 2 years. And for instance, on the upper left-hand side, you see a pancreatic cancer patient. where they had a rather significant reduction in liver mets over a short period of time, and a mesothelioma patient in the center that had a rather significant reduction in mesothelioma. So we're seeing some anti-tumor effect. Again, it's based on these are the sickest of the sick patients. We're using messenger RNA. We're redosing on a short-term basis, and we're seeing some hints, if you will, of efficacy. One of the issues here is that with messenger RNA versus lentivector, with lentivector, when you give that drug to the patient, those lentivectors actually expand in the patient, and it becomes a living drug in the patient. When you give messenger RNA, those cells don't expand. If the cells expand, they actually split their payload, and so you never have a higher dose a week or two after administration. With messenger RNA, you're always given the top dose, so you're not concerned about a peak in terms of the therapeutic change in the therapeutic index. So, as I mentioned, we've been working with both groups at Penn and the National University of Singapore. We have 7 cleared INDs. Most are in solid tumors. Excuse me. And those trials that have an asterisk, we have some interesting clinical data showing some anti-tumor effect. And again, these are very interesting programs. One is CD16, which is the basis for the Unum work that is the Unum company. Triple-negative breast cancer with c-Met, pancreatic cancer with the mesothelin antigen, mesothelioma, and GD2 and neuroblastoma. So we asked the question. We said, okay, what if we could really do something dramatic with this technology? And what if we could, in addition to increasing the safety because we didn't have the on-target off-tumor effect, but we're actually able to reduce the complexity because, again, we don't need to have the cells in suspension expansion mode in order to do the transfection. And we thought that this would do several things. One, it would allow us to go after solid tumor targets because we would be reducing the chance of toxicity on-target off-tumor toxicity. We'd be able to go after a number of cell types without expanding those cells and come down to what we think is a much more commercializable immunotherapy. And also, we want to be able to get these treatments to the patients within a few days versus a few weeks, which the clinicians were quite interested in. So we did the experiment, and we ran freshly isolated leukocytes, freshly isolated monocytes, and we loaded those with our technology and expressed them and compared those to expanded cells, and lo and behold, they react pretty much the same way. And so we followed our patents. We then began to think through, okay, how can we take this into the clinic? And in fact, we've been working with Johns Hopkins Hospital in Baltimore, one of the premier immuno-oncology groups in the world. And we began to look at the mesothelin antigen against tumor cell lines that express ID8. An ID8 cell line expresses mesothelin. You see a rather significant reduction in these cancer cells in this model. And then we had a rat model that was run at Hopkins, and we could see on a dose-dependent manner a rather significant reduction in those tumors that express mesothelin, and again, without any on-target off-tumor impact. And a standard Kaplan-Meier curve would show, again, some survival benefit using this technology versus a placebo control for these. And this is an ovarian cancer model we're using. So running out of time here, but we have issued patents on this. We have in vitro preliminary animal data. We have one patient that was actually treated at National University of Singapore using straight lymphocytes. And we're leveraging the 7 clinical trials that are going on at Penn and National University of Singapore. And we're about to embark on a next stage with Johns Hopkins to go into an IND at the end of 2016. And again, we believe this is a very novel approach, allows us to move into solid tumors by better managing on-target off-tumor toxicity, reducing time and complexity in manufacturing. And we could also use this technology as a plug-and-play. Mesothelin is the first target, but basically any antigen target we can use and will use as we partner this technology broadly. And because the technology has multiple uses, we can actually load in checkpoint inhibitors, which we've begun to do the work at Duke. So thank you for your attention, and look forward to answering any questions you may have after the presentation. Thank you.